welcome back to class. I just want to still remind you not to forget subscribing and sharing the video of these topics we are taking. We want to complete the aspect of photosynthesis, and at this aspect we'll be looking at importance of photosynthesis. We also look at evidence of photosynthesis in this short class. What are the importance of photosynthesis? So we'll start with that. Number one, photosynthesis helps in the purification of the atmosphere. In what way? Carbon dioxide is the main raw material for the process of photosynthesis, and carbon dioxide is also referred to as the byproduct gas process of respiration. So when we have too much of carbon dioxide in the atmosphere, human and other animals will suffocate. But in this case, the atmosphere is purified by plants by eliminating this carbon dioxide and using it to produce glucose. Number two, photosynthesis helps in the production of organic material. Helps in the production of organic material in form of glucose. So production of glucose, which is the bedrock of every food that is obtained from plants. Production of glucose, which is the bedrock of every food that is obtained from plants. Number three, it also helps in the beautification of the environment. As glucose is being produced, plants will grow and the environment becomes more beautiful as a result of these plants in the environment. What happens to glucose after it is manufactured in the leaf or other chlorophyllous part of the plant? Number one, things that happens to fate, I can put like fate of glucose after it has been manufactured. Number one, it is stored, can be stored as starch in different parts of the plant, depending on the type of plant. Can be stored as starch. Number two, glucose could be combined with elements such as nitrogen, glucose plus elements like nitrogen, phosphorus or sulfur to produce protein. So glucose can combine with nitrogen, phosphorus, sulfur to produce what we call protein. So the proteinous food that we have has its bedrock or its backbone as glucose. So when nitrogen, phosphorus or sulfur, either one of them or combination of them combine with glucose, we have proteinous food. Number three, glucose could also be oxidized to produce ATP. Oxidized to produce ATP, and this number three is as simple as defining respiration. So glucose will be oxidized to produce ATP, which is the energy currency of every living organism. So that's the fate of glucose. Now, the evidence that photosynthesis has occurred in a living plant is actually tailored back to the presence of glucose in the leaf, to show that photosynthesis has occurred in any plant. What we are looking for is glucose present? Because once there is photosynthesis, what we are going to observe there is the presence of glucose. So evidence that photosynthesis has occurred in a living cell is actually the presence of glucose in that in the chlorophyllous part of that particular plant or organism. So we'll be looking at how to test for starch or glucose in leaf. Test for starch, that is evidence. The evidence that glucose that photosynthesis are called in a leaf is actually the presence of starch in the leaf or in the chlorophyllous part. We shouldn't forget that it is glucose that combine a large number to form starch. In fact, the monomer of starch is actually glucose. The monomer of starch is glucose. When we eat our carbohydrate food, what we are expecting from this carbohydrate food or starchy food is actually glucose, because they are the constituent of this carbohydrate or starchy food. So I move on with this second part now, which is evidence of photosynthesis, which is something else. Just to say, evidence of photosynthesis can also be referred to as the presence or test for starch in leaf, or evidence of photosynthesis. So if you look at the equation for photosynthesis, we have glucose and oxygen as the products. So sometimes some want to look at the presence of oxygen, and that brings us to something else again. If I refer back to the importance of photosynthesis, aside from the purification, photosynthesis also helps to replenish depleted oxygen in the atmosphere. When the oxygen in the atmosphere is depleted as a result of excessive respiration, photosynthesis will help to replenish the oxygen that has been depleted from the atmosphere. So the test for starch in leaf is actually the evidence. Positive test for starch in leaf is an evidence that photosynthesis has occurred in a leaf. Aside from that, the presence of or the release of oxygen gas could also be used to show that photosynthesis has occurred in a leaf. So how do we test for starch in leaf? The test for starch in leaf has some steps, and that will help us to look at other things like materials needed for photosynthesis. Test for starch in leaf, what are the requirements? We need leaf that is obtained from plants that have been exposed to sunlight for a good number of time. We also need iodine solution. Aside from that, we need alcohol or ethanol. Aside from ethanol, we also need water, then heat or source of heat, So aside from these ones that are going to be used up, we also need beaker and slab to test for the presence of leaf in starch. What do? We get leaf. We pluck leaf from plants that have been exposed to sunlight for a good number of hours. Green leaf. After it had been plucked, so the first thing is just green leaf from plants exposed to light for more than 5 hours. That's the first thing we should keep in mind. Then once we get the leaf, the next thing is to boil the leaf in water. Boil it in water. Allow it to boil. Why are we boiling the leaf in water? It is just to burst the starch granules. Because of the starch granules. This is a small capsule keeping or housing the starch that might have been formed or stored as a result of the photosynthesis. So once we burst the starch granules, they are exposed. The starch are exposed. Then after we have burst deposited the starch granules. The next thing is to insert the leaf in ethanol. Insert the leaf in ethanol. Insert the leaf in ethanol. Why do you insert the leaf in ethanol? The insertion of the leaf in ethanol is just to decolorize the leaf, just to remove the green color. After that, the ethanol has the ability to shrink or to break the leaf. The next thing to do is to quickly dip the leaf in warm water to prevent the leaf from breaking. Dip the decolorized leaf in warm water to prevent breaking. Then the last part is just test for the starch in the leaf. So how do you do that? You place the leaf on a slab and add 2 to 3 drops of iodine solution. Once you add this, if the color of the part where these drops have been placed changes to blue-black, it is an indication that glucose is present. So what we are expecting is we have observation now. Observation, color change occurs. That is to blue-black on the leaf. And the conclusion is that photosynthesis, the conclusion is that glucose or starch is present in the leaf. And starch will only be present if glucose has been formed in large quantity to form what is called starch. So the conclusion will just be starch or glucose. Starch is present and photosynthesis had occurred. Now, this particular experiment can lead us to so many things. And what do we expect from this experiment? This experiment will make us show those things that are needed for photosynthesis. Things that are needed for photosynthesis.